All right, so today is Wednesday. I want to welcome everybody again to the Technology Ideations Innovation Ecosystem online platform forums. This is the Open Ideation Forum. Our focus here is to come up with new applications to existing solutions that are going to be presented here, and also to listen and understand what it is that they're trying to accomplish and identify where it is that we can assist them in achieving their goals, and also if their systems or their solutions apply to your field of study. Today we had Silver Thread, Biosortia, and AVOT that we're going to present. I have not seen Silver Thread on here for those of you that are here. So we're going to start off with Biosortia. Fascinating technology here. A lot of good applications across different industries. So with that, I will turn this over to Ross from Biosortia and have him take over.
Terrific. Thank you. Thank you, Eugene, and thank you for allowing us to present this. It is an interesting technology, something that people never really thought would be possible. So as we go through this, I hope to get some great questions on this, and look forward to letting everyone know that we've got 3 presentations total. The first one's already recorded, and that's available at Eugene's site, Technology Ideation. The second one we're presenting today will end up also at Technology Ideation's site. And then we'll do a third one. The first one was on essentially energy, and let's go to where we are. Here we go. It was essentially on materials, defense, and energy. The one that we're doing today is on therapeutics, health, and nutrition. So therapeutics, health, and nutrition will be covered today, and the ideation around that. And then on the 27th, we'll cover sustainability, the environment, and agriculture. And it's easy to say, why can a technology be so broadly impactful? Same thing with genomics. Genomics is a life science-wide impactful technology. Industrial scale microbiome mining is that same type of broad scale impact, except for it's going to be able to deliver the molecules that has been hidden from all other technologies. And we'll talk about that just a little bit. So in the first presentation, we did cover part of the reasons why, and I'm going to go through these slides rather rapidly with the hope that we can put some context together, but individuals will go to that first presentation for a little more details. So in the first presentation, we basically presented why life on Earth has evolved and how microbes have been evolved and how they ended up virtually getting into all organisms, including the human, including our entire food web, and how they evolved so rapidly. And we're going to play a video today that if you've never seen this video, you're going to be quite surprised at what we describe as the calculating power of microbes. And we also discussed some of the existing technologies that have been around over the years, including what worked and what hasn't worked as it was projected. So there's the reality that when we look just at therapeutics, 50% of the drugs on the shelf have a connection to microbes, either directly, indirectly, or inspirationally. And when you look at how productive that's been for what limited access has been and what limited dollars have been spent on that technology, it's a pretty productive technology. As we started to go to indirect technologies, the productivity went down, the promises went up. The promises haven't really been fulfilled, but a lot of dollars and time is going into those technologies, which, interestingly enough, is important because it really facilitates all of the opportunities to benefit now from industrial scale microbiome mining. You said there was a chicken and egg. It's certainly one could have come first, but having all the technologies, all the toolboxes are important. So what we'll talked about in the first presentation is what we do and where we go to get it, and how our business model itself works. We're at essentially the center of a spoke and hub system where all of the spokes can work with us to get these molecules to drive pipeline products, not only into therapeutics, but into a wide variety of life sciences. Then we talked a little bit about our current scale, and we're going to talk about that just a little bit more, but there is details about that in that first presentation. What we're going to start to talk about in this presentation is the connection between the disease, health, and nutrition of the human microbiome. The evidence is mounting nearly daily. In fact, I just did a brief look at Google Scholar and sorted based on dates. And what it appears like is the scientific papers that are published on the microbiome itself is doubling every 3 years. So, and that's been since 1990. So this is a tremendously advancing field with the recognition that the human microbiome is impactful across virtually every disease state and some of the diseases we would have never expected. So one of the things about precision diagnostics, the role of genomics and biochemical fingerprinting for habitats now and in the future, is we've learned that genomics itself is not the complete picture. It's a part of the picture that can provide guidance. But the BRCA gene itself is good, solid evidence that it's predictive to a certain degree, but there are so many things that are involved with just the genomics that would suggest or predict a higher probability of cancer for having that BRCA gene. Right now, it's a high probability, but it's not 100%. So the question comes up, why is there such a complexity to it? Why is the genes alone not enough? And certainly 30 years ago, there was the belief that understanding the genes, we really would have that complete picture. But that was before we really understood epigenomics. We had no idea about the microbiome metagenomics and the microbiome in the human having such an impact. And certainly when you talk about any organism, any habitat within that organism, you're talking about the biocomplexity. So it's not just the neat genes, it's what is resulting from all of that. What are the actual molecules, the proteins, the enzymes? How do you get a better picture of predictive capability? And right now, we don't have direct access to so much of that information. And that's one of the biggest challenges. But advancing existing tools and utilizing the new capability of industrial scale microbiome mining, we fully believe the dots will get connected, especially when you talk about predictive fingerprinting of habitats, whether that's a human, whether that's agriculture, whether that's sustainability, whether it's threat detection. There's going to be the capability to understand in a way we have no way to understand today. I'm going to use this presentation to show you just exactly how fast a single species, E. coli in this case, can adapt and change. So we'll play that.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, of course, thin agar, that bacteria can move around it. The background is black because they're incontinent, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right, resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it 10 times as much antibiotic. And you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So I think that's one of the most interesting videos talking about the power of a microbe to adapt to its environment rapidly. In 11 days, it outcalculated the toxin that was designed to kill it at every single level. What's important about this is we know in science that microbes have short life cycles. We know that they adapt to their environment dramatically. We also know we can use microbes to do things for us. So if we were to insert genes to produce a particular product, we might choose E. coli to insert those genes and produce that product. But with horizontal and vertical evolution, the microbes themselves, whether they're in us or they're in other habitats or environments, can outcalculate virtually any kind of computational calculation rather rapidly. And that's one thing to pay attention to, because if we're trying to figure out what microbes are doing from calculations alone versus really understanding what microbes are doing in specific habitats and what's resulting from those microbes, we're only getting a fraction of the picture. So let's look at it a little bit differently when we talk about industrial scale microbiome mining, because I've had many people say, this is hard and it's impossible to do. And I think people that are at a lab benchtop and they think about a typical sample size, it's certainly easy to say what we're talking about with industrial scale microbiome mining has never been taught or practiced other than the proof of concepts that Biosortia has done. So let's just look at a different kind of process. Let's look at gold mining. Hard rock gold mining, for instance, if you have 1 million grams of hard rock, whether that's quartz or granite, and it has 4 grams of gold out of that million, it's economically viable to go through that processing in order to obtain that gold. That's kind of incredible, since 4 grams is going to deliver you about $250. But the technology is there. The technology has been improved. The scale has been improved. When we talk about industrial scale microbiome mining, to get at the hidden small molecules in microbiology and biology, it's just never been done. And what we've got to do is start that process to improve that process, to deliver the products. And we know from history, microbes have delivered 50% of the drugs on the shelf and other products throughout life science. So obtaining microbes and microbiomes at the quantity and quality while utilizing existing and advancing technologies will prove critical to fingerprinting any habitat and discovering next generation starting points for valuable products across all of life science. So let's talk about the scale, and let's talk about the environmental friendliness of it. What you see here is 8 Olympic-sized swimming pools. And where do we go? We go to the aquatic habitat because, as you'll learn when you watch the first presentation, is that microbiomes of the world overlap dramatically, including the human microbiome in genes, metabolic pathways, and the small molecules produced. But what we're talking about is how do we get at those hidden sub-peak molecules that are in parts per trillion and less quantity? You've got to start with a scale. And many will go, this is incredibly complex. And I'm here to say, just in our proof of concept, we've shown that it's not that complex. So we're talking 8 Olympic-sized swimming pools in order to get at the hidden small molecules. Now, rightfully so, that is much, much larger than the typical lab sample of 100 milliliters, which is shown in the center of this slide. We're talking 200 million times the size. But why I'm showing it here is it's not that big of a stretch to do what we're talking about. And again, this technology is at day one. So when we look at how deep we need to go to understand the molecules that are hidden in the microbiome, we can look at the human microbiome and what drug-like activities, when we put molecules into the human and it has an activity, what kind of, let's say, parts per billion is that? Knowing that the metabolite density of an aquatic environment is dilute compared to the human metabolite density. So that gives us guidance on how deep we need to go. And that's why 20 million liters at a particular density is a good starting point for getting virtually the vast, vast majority of these hidden small molecules. Now, if you go deeper, which is economically viable and environmentally friendly, completely environmentally friendly, then you start getting into those sub peaks of those molecules. But in a human, you're talking about anywhere from a few hundred to a few thousand parts per billion is what a drug would generally work at. And that's exactly what we're trying to do, is figure out what we can see by going at that density. And what we see is we see about 80,000 addressable small molecules from a single aquatic microbiome. We also see that it would be very easy to do 10x that, meaning that we're ending up with 1,000+ kilograms of the metabolites that are directly involved in cellular signaling of a microbiome.
Hey, Ross, can I stop you there for a second?
Please.
So logistics for a minute here. So you're not actually transporting millions of gallons of water or anything like that. I think that needs to be addressed here if you're not.
That is a terrific point, Eugene. What we do is we go from the density of the existing habitat and rapidly and effectively in quality and quantity without harming the microbes, concentrate it, and then go into preservation mode and do that very rapidly. So we're collecting just the microbes in a non-chemical process from virus size to 100 microns. So we're collecting a microbiome. Is there things in there that we'll also collect that are not technically part of the microbial biomass? Yes, there is. But the vast majority of what we collect, or what we design to collect, is that microbial biomass. And then we start preservations from the time we remove it. From the time we remove it to the time we start to preserving it, or the start of that, is 2 minutes and 14 seconds. It's been dewatered from what could be 0.02 to 0.05 grams per liter to 200 grams per liter. So the vast majority of the water is removed at that point. What we see in this particular chart, or in this particular page, is that we have peer review, and we've got a number of peer reviewed papers. But what's important about this paper is it laid out a proof of concept of around 370,000 liters that led to essentially the metabolites of around 2.5 kilograms. So it does take a lot of material to obtain the quantity of material to find things. And when you look in a typical aquatic biomass or a microbial biomass, you're going to get a wide different ranges of proteins, lipids, metabolites, polysaccharides, nucleic acids, and the inorganic materials. It just depends upon the environment you're going. But what we've been able to prove is we could get at unknown, novel, bioactive small molecules that we could discover. And that's what the cyanobufilin molecule is. And we'll have a reference to this scientific paper. It's already on the Biosortia website, as many other references are. Now let's look at one of the other issues, because one of the things I have run into to those that have studied a particular field, whether it's genomics or chemistry or whatever they may do, is they say, yes, but natural products.
Ross,
Oh, yes.
Sorry. Can I just ask one question? Does this system yours work for mining tailings also, tailings of mining, like with the solids, not just or just in water environment?
I think if all you're trying, I think if all you're trying to do is remove the solids that are at small densities within a tailings pond, and you're not trying to get the living, breathing microbiome, there are existing wastewater technologies that work better than this. This would be more expensive to obtain that kind of material out of a tailings ponds, but this is ideal. This technology is ideal for collecting the microbes and salvaging the microbes. So it would depend upon the purpose.
Yeah, but how do you deliver the microbes closer to the grains of gold, for instance, or any other metal that you want to recover from the tailings?
Well, the tailings may also have dissolved material, and this is not designed to obtain the dissolved material. The dissolved material will come right through this system, which is ideal for collecting microbes. We're only trying to get between 0.02 microns and 100 micron material in our system, which happens to be where most of the microbes lay.
Okay, thanks.
You bet. And I can get offline at any time to talk about that a little bit more if you'd like. So if anybody, please reach out to me and connect to me on LinkedIn. So one of the problems with natural products, and especially microbial natural products, has been there has been this dialogue that has bashed it for 40 years. And what I'm here to say is, with industrial scale microbiome mining and the existing technologies that have really advanced, and new technologies that are on the cusp, if not already impacting it, and those new technologies are machine learning and AI, which are developing rapidly, there is not one issue: complexity and diversity, isolation and purification, limited quantities for research and development, difficulty in identifying and validating targets, supply and sustainability concerns, or even high attrition rates that have not been improved or eliminated by combining technologies that exist today and utilizing industrial scale microbiome mining. The high attrition rates get eliminated, quite frankly, because you're starting with microbial products. Even when you derivatize them, improve them, optimize them, whether you're doing that chemically or computationally, you are starting with a molecule that already works at the cellular level. And with technologies today, you can calculate opportunities for, let's say, reducing the priority of molecules that you think could fail in preclinical or clinical. So we think all the issues that everyone will bring up as to why natural products don't work are actually the reasons that synthetic biology the reasons that genomics and even combinatorial chemistry were all talked about as being the next great provider of therapeutic opportunities. So can't have it both ways. In the original concepts around genomics is we were going to find out what these molecules did and then be able to put them to use. Well, if we can go get them directly and easier, then the same applies. Is putting them to use matters, and that's what industrial scale microbiome mining does. So we talk about this process in the first presentation, but what I want to talk about here is how everything gets improved with industrial scale microbiome mining and enhancing with AI. So the 5 ovals in the middle of the screen: spectral matching, activity prediction, structure prediction, target matching, and computational derivatization are all opportunities for machine learning and AI. One example is on the spectral matching, going to our unique and unknown compounds, we were able to increase the number of known natural indoles effectively through understanding our unknowns with AI and spectral matching. So indole is an interesting molecule because it's highly bioactive. There are drugs that are based on its substructure already, and it's an opportunity to just understand the wealth of diversity of molecules and opportunities that lie in these microbiomes that can be recovered. And when you look at synthetic chemistry, synthetic biology, indirect omics in general, being able to connect the real data is going to advance all AI dramatically. Right now, we're running about 10 million to 1 synthetic data to real data in drug discovery. Now, if you focus just on the real data, you've got a much higher odds. But the reality of it is synthetic data is so overwhelming. And we know, quite frankly, that randomizing the capability to find molecules fails miserably. And that's what combinatorial chemistry taught us. So really having the right data at the right place at the right time is critical to advancing all of life sciences, let alone advancing my slides. There we go. So I want to talk just a second about fingerprinting. And these slides will be made available as well. But when you talk about fingerprinting, whether it's the human or a habitat, or whether it's a threat detection, being able to understand the actual molecules is critical. Understanding the genomics, as I talked about earlier, gives you a fraction of the needed picture. So when we're talking about understanding what is actually being produced, you understand the metabolic pathways, which leads to the genes, but the genes themselves may not necessarily lead to the molecules unless we actually align them. And I think that that's the kind of thing that people have learned over the last several decades, is that there's so much we don't know. And in fact, in the human, there may be 100,000 plus small molecules and enzymes that are basically part of our biochemistry that we have no idea what they are. And through using the surrogate of the aquatic microbiome, our ability to uncover tens of thousands of those unknown molecules and be able to identify what are those blips in a mass spec, that's the opportunity you have. And that kind of fingerprinting can lead to predictive capability that's unparalleled. So let's look just an example at a little bit of the data we have. So what we're seeing working with Los Alamos National Lab is that one microbiome we processed ended up having nearly 80,000 compounds with enough information that we were able to identify nearly 6,000 of those compounds themselves. Now, the vast majority of the compounds fit within that Lipinski rule of 5, which is drug-like compounds. The other thing is when we use just analysis directly, out of those 6,000 compounds were 948 FDA-approved molecules. That does not necessarily mean drugs. 764 human blood metabolites, and 14.5% of the human metabolomes database that is microbial in origin. So there is an overlap pretty dramatically there. And if we were to look just at that 14.5% of human microbial sources and discount that to 70%, it's suggesting we will find, and sorry, this got in the wrong column, 240 potentially approved drugs out of one aquatic microbiome. Now, when you talk about mining and you talk about an opportunity, that kind of opportunity to find what could be billion-dollar molecules suggests that industrial scale microbiome mining should start tomorrow. Now, what kind of molecules did Los Alamos National Lab actually find from the known ones? Again, just like we saw with the scientific papers, it covered a wide range of probabilities and opportunities. And within that, it is very possible we found molecules that were in the waste stream, and it's possible that some of those weren't. So using just antivirals, 70% of the antivirals we found are not even prescribed. So essentially, that's where the 70% number comes in, is it seems like a good number for us to use, assuming that all of what is prescribed was contamination. Now, there's the likelihood that that's not the case, but that's why we use those numbers that we did. Now, here we get into the future, and one of the reasons the ideation is around, which is what in the world could come from a technology like that. Well, I think from our very first slide that showed the studies that are going on, there's hardly an area in therapeutics that will be untouched by industrial scale microbiome mining and the combination of indirect omics to advance these technologies along with AI. All of these areas, whether it's antibiotics, antifungals, antivirals, metabolic, cancer, personalized medicine, wound healing, obesity treatments, mood and mental health, allergy and asthma. Virtually every area will be touched by this. And that's the important thing to consider. And this will, like I've mentioned, these slides will be available. If you're talking about the health possibilities, certainly health possibilities and therapeutics run hand in hand, so there's going to be some overlap. But what about energy metabolism optimization, sleep regulation, mood stabilization, enhanced nutrient absorption, stress resilience, athletic performance, immune system modulation, skin and beauty, hydration and electrolyte balance? Again, the opportunities from understanding the cellular level, and the cellular level is the microbiome level, the microbes, as a key contributor to that cellular understanding and cellular signaling. You're talking about health possibilities that are wide-ranging. And the same thing with nutrition. Again, therapeutics, health, nutrition, there's tremendous overlap. Let's just pick a couple of these. Optimized plant-based nutrition, food intolerance management, nutritional deficiency prevention, protein quality improvement, antioxidant production, alcohol metabolism. When you talk about the possibilities, they are wide-ranging. Now we are ready for questions, and I look forward to that. So thank you for the opportunity to present.
Well, thank you, Ross. Good presentation there again. Appreciate you being here. And so now when you talk about these applications in areas such like, cosmetics, and which is a huge industry, so we'll just stop with that one there. What how would you broach that? How would you be able to collaborate with someone or with a company in that industry?
What we would look at is the spoke and hub system. So Biosortia focuses on what nobody in the world is doing. We're going to get those molecules, understand what those molecules are, make them available for testing. And that would mean that we've got to do that mining at the scale and quantity and quality to bring these molecules forward. Now, we estimate that out of the microbial world, we'll be able to bring directly hundreds of thousands of microbial molecules. Once we do the derivatization of those, we'll be talking about millions of microbial sourced cellular signaling molecules that already work in nature. We're talking about multiples on top of what already exists today. 50, 100, 500 times the libraries that exist today. Only we'll have the kind of categorization of these molecules that we'll understand. Now, that's not going to be day one. That may be day 1,000, where we really have improved all systems and capability. But the ability to impact all these industries we've been talking about will be wide ranging because of those spokes. We want people in the cosmetic world, in the therapeutics world, people trying to find molecules that can impact cancer and impact other diseases. And we want them to come to us and create a relationship with us so we can work with them, so we can get this molecules and this intelligence to them. And even those that just want data, what molecules we have, how they work, how they connect to the metabolic pathways and the genomics, we want to be able to work with them as well, because one of the big challenges for everybody out there doing drug discovery is, again, the fact that there's very little real data to go from. And no matter how you look at computational work, the better the data set, the better the results. And that's one of the things that we're going to strive for, is creating the best data and the best molecules and making them available to our partners and our collaborators.
Okay. So let's open it up. Does anybody else have any questions for Ross? Chime in. The floor is open. If you're trying to talk, you're muted. All right, then I will continue on. So you talk about humans and about health benefits. What about agriculture? I mean,
It's going to have a
obviously,
substantial impact in agriculture, Eugene. Right now, there is hardly a new patent that has been approved. I think that 98% of the agriculture patents are off patent. There's serious issues with the products that are on the market and the fight to get those products off the market. There needs to be a higher level of focus and specificity, and then there needs to be able to come up with alternative approaches that work with the adaptation of the organisms that are targeted. So to me, understanding the microbiome is going to be key to agriculture's future.
okay, yeah, because here at this summit, I actually talked to somebody that was working on gene editing, which rather than gene modification, which it is modification, they're removing susceptible genes rather than adding prohibitive genes, and apparently that's treated differently or looked at differently from a USDA perspective. So I'm hoping to get them on the forum later to present on what they're doing.
Yeah, and I think what's interesting is that when you talk about understanding the microbiome, the way our depth of discovery will be, gene therapy, whether it's for agriculture, agrochemical, whether it's for therapeutics, there is a number of opportunities to advance that technology dramatically and rapidly by the understanding we can uncover.
Okay, then I have one more if nobody else has anything. And that is, so once we harvest, we classify, identify, are we going to continue to go out and harvest, or are we going to try and synthesize these? What
The
would be the
goal
next
for
stage?
GMP is synthesis. Interestingly enough, having an access to what will, at least at first, be a inexhaustible supply could open opportunities for direct. One of the things we're firm believers of, standards may come directly from this source because the quantity needed worldwide will be grams rather than, a kilogram. So it may be that standards for hundreds of molecules we don't have standards for now will come from the microbiome. So there could be a massive new opportunity to make sure that aspect develops, and it may end up being well more than 100. It may be thousands of standards could be created by access to microbiomes.
All right. Well, thank you. One last time for questions before we move on. Another phenomenal presentation. I appreciate it. It's a fascinating topic. Something I'd never been exposed to until you reached out to me, and I've been reading about it. So appreciate you being on here, and you're going to be on again in a month. And
Be on again in a month. And the one thing I'll say is I found it interesting because Biosortia's industrial scale microbiome mining is pretty much unheard. But going to ChatGPT and asking questions about it, I'm almost constantly surprised at how well it answers those questions, just to let people know.
All right. Well, thank you very much again.
Thank you.